Hey everyone! In the last video you learned how to name chromosomes. In this video we'll go over how to count chromosomes, DNA molecules, and gene copies as a cell proceeds through cell division. This will become important once we look at mitosis and meiosis in detail. And in this video we have just one question. And our question is, how do the number of chromosomes, DNA molecules, and gene copies change as a cell proceeds through cell division? Now that you know how chromosomes change and how to name them, let's determine the number of chromosomes, DNA molecules, and the number of copies of each gene in our cell at different points during cell division. To make everything easier to see, I didn't draw the nucleus in these cells. Also, I've given each chromosome type a different color, but if these were real chromosomes, we could identify each chromosome type by size, centromere location, and band pattern. First, try to fill in this table on your own in the learning guide. Remember that the person who does the work does the learning, so even if you get some things wrong as you fill in this table, you'll remember this better than if you just wait for me to explain it. And second, write down the ploidy of this cell. I'll give you a second to pause the video. Okay, so for those of you who filled out the table, you are an excellent student. We can tell that this is a diploid cell because before replication, there are 2 of each chromosome type. So this species is 2N. We have 3 types of chromosomes, so N equals 3. Since we have 3 types, and 2 of each type, we have 6 chromosomes total. Of course, we could also just count these chromosomes: 1, 2, 3, 4, 5, and 6. Before DNA replication, each of the 2 chromosomes of each type consists of only one DNA molecule. We therefore have 6 DNA molecules in this cell. Recall that 2 chromosomes of the same type have the same genes, and chromosomes of different types, or non-homologous chromosomes, have different genes. Again, these genes and the chromosomes they are on have not yet been replicated, so we see 2 copies of the A gene, 2 copies of the B gene, and 2 copies of the C and D genes. So at this point, there are 2 copies of each gene. After the chromosomes are replicated and subsequently condensed, each newly made DNA molecule remains attached to the original at the centromere. So these 2 attached DNA molecules are considered one chromosome. Since we started with 6 DNA molecules, and each one was copied, we now have 12 total DNA molecules. Since there are 2 DNA molecules in each chromosome, there are still 6 chromosomes total. As for the number of copies of each gene, when each chromosome was replicated, their genes were also replicated. We see that there are now 4 copies of each gene in this cell. When the sister chromatids are pulled apart, each chromatid is now considered its own chromosome. So there are now 12 total chromosomes in this cell. The number of DNA molecules and the number of copies of each gene don't change. All we've done is double the number of chromosomes because each sister chromatid became its own chromosome. After the cell splits, the 2 daughter cells now once again have 2 copies of each chromosome, for a total of 6. Each chromosome now only has 1 DNA molecule, so we have 6 DNA molecules as well, and we have 2 copies of each gene, which is what we started with. Okay, so at this point you should be able to count the number of chromosomes, DNA molecules, and gene copies in a cell at each point during cell division. If you can't, as always, feel free to watch the video again. Now, in the next video, I'll give you a brief overview of mitosis and meiosis. See you then.